Hello, everyone. Welcome back to my video tutorial for the Maxfuse package. From this video, I'm going to show you how to perform CODEX data and single-cell RNA sequencing data integration using the Maxfuse package. If you didn't install the package, you can use pip install Maxfuse to install the package. Then we know the other packages here for CODEX data and single-cell RNA sequencing data integration. For today's tutorial, you can download the data from this link. I downloaded the data already. We can have a look at the data in my folder. Let's have a look in my folder. You can see in this folder we have the tonsil CODEX data, tonsil RNA data, the counts, RNA names, and the metadata. I believe you know how the RNA data looks like because I have a lot of videos about the single-cell RNA sequencing data analysis, but I only have one video about the CODEX data. We can click the data and open it. So you can see in the CODEX data, we have the cell IDs, hotshots for the nucleus staining. Then in this data set, they performed the multiplexed data collection for 46 proteins. You can see the protein name from CD38 until E-cadherin. So the expression matrix already normalized and scaled in this dataset. So here are the expression matrix data. Then we have the cell cluster M here. So you can see the cell types for each cell in this column, and also we have the centroid X position and the centroid Y position. Then we can use the X and the Y position to recreate the image. So now you know the data information in the CODEX dataset. Then we can have a look at the Nature Biotechnology paper again. You can see in the figure 1, they have the overview of Maxfuse pipeline. They divide the analysis in 3 stages, stage 1, stage 2, and stage 3. I will use the CODEX data and the RNA-seq data to demonstrate the analysis for 3 stages. But for today's analysis, we are going to pre-process CODEX data and the RNA-seq data. Basically, today's analysis will be in this box. You can see here they name the one modality as Y, the other modality as Z. So we can consider the Y as CODEX data and the Z as RNA-seq data today. So for data preparation, we need the original CODEX data and the RNA-seq data, and also we need to prepare the linked features CODEX data and the RNA-seq data. The linked features means we are going to prepare shared features for each dataset from the CODEX and the RNA-seq data. So at the end of today's video, we will get a full dataset, the original data and the shared data. So now we can go to Python and do the analysis. So the first step, we are going to load the protein data. I showed you the protein data is in the tonsil CODEX CSV file. So we can just read the protein data in as protein. Let's run. Then I showed you the centroid X and Y position. We can use the cell position to create a tissue image. Okay, you can see we created the image for the CODEX data. You can see there are 6 different cell types in this dataset. T cells and B cells in proliferation, CD4 T cells and CD8 T cells, plasma cells and dendritic cells. Because we noted all the data from the CSV file for the CODEX protein data, but for the analysis, we just want to get the expression matrix data for all the proteins. I showed you we have 46 proteins from CD38 to E-cadherin. So we can list the proteins name here, then subset the data from the CSV file and create a protein A data. And we set the proteins names as the column variable names. So let's run this code to create the protein A data for today's analysis. I also showed you we have a column called class term in the CSV file. This column contains the cell type information. Then we can add the cell type information in the protein A data. Now we can have a look at the protein A data. You can see in this data set we have nearly 180,000 cells and 46 proteins. And at the moment we only have one observation about the cell type. We can have a look at the observations. You can see the row names are the cell IDs. Then at the moment we only have the cell types for the observations. And also we can have a look at the variable names. You can see near the protein name that we set as the column name. So next we can read the RNA data in. For the RNA data, we have the RNA count text file and also we have the RNA names in the RNA name CSV file. Then we can load the data and create a RNA data, and we set the RNA names as the column names. Let's run. Okay, we created the RNA data. We can have a look at the RNA data. You can see in this data set we have nearly 13,000 cells and more than 33,000 genes. We also have the RNA metadata. Instead of doing all the analysis, we can just load the RNA data and get the cell type information. Then we don't need to run the scRNA analysis. So now we load the CODEX original data and the RNA original data. Next, we can construct two shared arrays, the RNA shared and the protein shared. In both arrays, the columns have the same gene name. The matched features in two shared arrays can be used as weak link features to obtain the initial matching during the initialization for the Maxfuse analysis. You may realize the problem here because the column names in the RNA data are gene names, but in the protein data, the column names are the protein names. If we want to perform data integration, we have to convert the column names to match RNA data and protein data. Because we only have 46 protein data here, so we can convert the protein names into gene names. That will be easier. So when you download the data I showed you in the beginning using that link, you also get a protein gene conversion CSV file. We can have a look at that file. You can see we go up. Here we have the protein gene conversion CSV file. We can open it. You can see in this file, we prepare the gene name for more than 200 protein names. In most cases, you can use this CSV file for analysis. If you have different tissue, you may have different protein names. You can add your gene names for your protein name in this data list. Then you can use this CSV file for your analysis. So now we can load the conversion CSV file in. And I already showed you the data in the CSV file. We have the protein name and the RNA name. So the code here basically is to get the gene name for each protein name in our CODEX data. Let's run the code. Then we can have a look at the RNA protein correspondence file. You can see now we have the array for the protein name in the CODEX data. The protein names are in the second row and the gene names are in the first row. If you have a look at the details in this file, we only have 46 protein names for the CODEX data, but we have more than 46 gene names here. You can see because, for example, the CD8 protein name, we get the gene name as CD8A and CD8B in the RNA-seq data. For the CD3 protein, we also get CD3D and CD3E gene name from the RNA data. So in total, we have 53 gene names here. So now we can use the RNA protein correspondence file to subset the data. First, we can get the shared RNA data. Then next, we can get the shared protein data. Then we can print the data shape for both RNA shared and protein shared data. Let's run the code. You can see now we got the RNA shared data. We have the same number of cells as the RNA data, but now we only have 53 genes. For the protein data, also we have the same number of cells. Now we have 53 gene names for the protein data. Because we are going to use the protein shared and the RNA shared data to perform the initial data integration. Then we want to make sure the shared features between RNA and protein shared data are variable features. We can set the threshold for both the RNA shared features and the protein shared features. Then we subset the RNA shared and the protein shared data again. After subsetting the data, we can print the RNA shared and the protein shared data shape again. Let's run. You can see now, after subsetting, we still have the same number of cells, but now we only have 32 genes for both datasets. So we have the RNA shared and the protein shared data. We can perform a standard scRNA processing steps to connect the cells for both RNA and protein data. I showed you the CODEX protein data was normalized and scaled already, so we can skip those steps. We can just run k nearest neighbors, then UMAP, because we have the cell type information. Then we can plot the UMAP, use the cell type information. Let's run. Okay, you can see we got the UMAP cell clusters. We have 6 cell types here for the CODEX dataset. So next we can run the standard scRNA processing steps for the RNA shared data. We are going to normalize the data, then log transform it, then scale the data. After that, we identify the neighbors, run UMAP, then plot the cells according to the cell types we got from the RNA metadata. Let's run. You can see we quickly generated the UMAP for the RNA data. Again, we have 6 cell types here. Here we cluster the cells based on the 32 genes that are shared with protein data. So we processed the protein shared and the RNA shared data. Now we can get the two arrays. One is the RNA shared and the other one is the protein shared. We will use the shared RNA and the protein arrays for the Maxfuse analysis. Let's get the two arrays. So after we got the two shared arrays, now we can analyze the original sequencing data. First, we can analyze the protein data again. As I mentioned, we don't need to run the normalization, logarithm transform, and scale the data. We just need to run the k-nearest neighbors and the UMAP. And we use the cell type information from the original analysis. Now for the original sequencing data, we have 46 proteins. Let's run the analysis for the original sequencing data. In the Maxfuse tutorial, they call this original data as active protein measurement and active RNA measurement. So let's run the active protein measurement now. Okay, you can see we generated a UMAP for the CODEX data again. Here we cluster the cells using all 46 protein expression matrix data. Again, we have 6 cell types here. So next we can analyze the active RNA measurement. We can normalize the data first, then log transform the data. Here we are going to identify highly variable genes for the RNA data. Normally we use 2000. For this demonstration, we are going to use 5000 variable genes. Then after that, we will subset the RNA data, just keep the variable genes. Because there are no features in the protein data, Maxfuse doesn't require all the RNA features for the data integration. This is also the reason that we didn't run this analysis before the RNA shared array, because here we subset the data. We are going to run data scaling on the highly variable genes. After that, we can run the nearest neighbors. Then UMAP, and we plot the cells using the cell types information from the RNA metadata. Let's run. Okay, you can see we generated the cell cluster now. If you compare the cell cluster here with the RNA shared, we have a much better cell clusters UMAP than the RNA shared data. Because here we use the 5000 highly variable features to cluster the cells. So now we analyze the both protein and the RNA active data. Then we can get the two active arrays, RNA active and protein active, for the Maxfuse data integration. And at the same time, we want to make sure all the features are available for both RNA active and protein active dataset. So let's get the two active arrays now. So now we should have 4 arrays: the protein and the RNA shared array and the protein active and RNA active array. We can use the code here to have a look at the data shape for each array. Let's run. You can see I showed you already, we have 32 features for both shared array and 46 proteins for the CODEX active array we only kept 5,000 variable genes for the RNA active array. So we will use 4 arrays to perform Maxfuse integration analysis. I will show you how to run the Maxfuse analysis in my next video. I'm going to stop from here. You can try to download the data and follow the video to prepare 4 arrays for next video tutorial. I hope my video can help your data analysis, and see you in my next video tutorial.